should distinguish two elements. So, of course, the follicles, and except the follicles, which are covered by the follicular cells, you should also remember about the parafollicular cells, which are called also the C cells. And from the higher magnification, what we have here? Of course, between the follicles, we have the connective tissue stroma. Between the follicles, because this is the endocrine system, that's why these red structures with flat nuclei, these are the blood vessels, these are the capillaries, sinusoidal or fenestrated, sorry, let's keep the version of the Professor Duszewska, because hormones which are produced by the follicular or parafollicular cells should enter directly into the circulatory system, okay? And here in the middle we have a nice follicle. They can vary in size and shapes. Inside the follicle we have the colloid with the thyroglobulin, and here we have the follicular cells. And depending on the activity, if the follicle is active, these cells are flat, they create simple squamous epithelium, and when the follicle is not active, we have simple squamous epithelium. When the follicle is active, we have simple cuboidal or even cylindrical epithelium. Okay, and what about the C cells? C cells or parafollicular cells. They can be located, of course, between the follicular cells. Sorry, but in this type of staining, we can't see them. We should do the immunohistochemical staining to see it clearly. But also they can be located between the follicles, somewhere here. And if you will see such place with densely packed nuclei, or the place which looks like this, you have two options. First option, and I think it's probably correct option, that here also you have the follicle, but you have the place where you cut your follicle over here, just through the follicular cells, okay? So that's why we can see the lumen, because you cut these cells, these follicles, sorry, through the cells. So you have a lot of nuclei belonging to the follicular cells. But maybe also this is the group of C cells. We don't know, because in this type of staining and in normal routine AG staining, we can distinguish them. And over here we have another endocrine organ. This is the adrenal gland, which is covered by connective tissue capsule. Here we have connective tissue capsule well developed. And histologically, we can divide this organ into the cortex. So from the connective tissue capsule to this place, we have the cortex. And here we have the medulla. So the violet part, this is the medulla. In the center of the medulla, we have the vein. This is not the artifact, this is the vein anatomically located there, it's correct, okay? You have the blood inside, some endothelial cells, so for sure you have the blood vessel. And when we talk about the cortex, we can distinguish 3 or even 4 layers. So zona glomerulosa, just under the connective tissue capsule. Then we have zona fasciculata, and then we have zona reticularis. But if you see the pictures clearly, you will see that between the zona fasciculata and zona glomerulosa, you will see the densely packed nuclei. These nuclei belong to the stem cells. So we have 4 layers. Zona glomerulosa, exon, zona fasciculata, and zona reticularis. From the higher magnification, we can see some connective tissue elements from the capsule. This is the zona glomerulosa, and the endocrine cells here are arranged in arches and spherical clusters with numerous capillaries, sinusoidal capillaries. So each cluster is surrounded by the capillary. Okay, these are the endocrine cells, and these are the capillaries. Then you have this X zone with the stem cells, and then you have the most extended zone. This is the zona fasciculata. Here, blood vessels run straight, they do not have the appearance like arch, but they run straight. And along the blood vessels, the endocrine cells are oriented. And these endocrine cells from the zona fasciculata are called also spongiocytes. And probably you should know it. Spongiocytes because the cytoplasm is foamy-like. Of course, we can see the lipid droplets from this cell because the lipid is dissolved by using the xylene and the alcohols. We have empty lipid droplets. Okay, and let's move to the respiratory system. We will start from this slide. Do you know what is this? Is it trachea or bronchus, primary bronchus? What do you think?